Hi, this is a quick video lesson for Higher Human Biology Unit 3, and this is on the topic 3.6, which is specific cellular defences against pathogens. So let's get going. We are going to look at the role of lymphocytes. So at National 5, you were taught that phagocytes engulf and destroy pathogens, and we were taught that lymphocytes produce antibodies which destroy pathogens. That second part's not quite true. First of all, there's two types of lymphocyte, only one creates antibodies. Second of all, the antibodies do not destroy the pathogens. The antibodies lead to the destruction of pathogens by the phagocytes. So let's look at how that works. What is a lymphocyte? Well, it's a type of white blood cell which is involved in the specific defense against specific pathogens. So first of all, they have to respond to antigens on the surface of a pathogen. An antigen is a molecule on the surface that triggers this immune response. So these are normally proteins. They are on the outside of the pathogen. In fact, they're outside of every cell. But the lymphocyte has receptors that detect these antigens. And if it detects a protein that triggers an immune response, then that's called an antigen. So pathogens, the lymphocytes recognize as being foreign and being harmful because of the antigens they have on their surface. So here we have a bunch of different lymphocytes, and you can see there's differences between them. Each lymphocyte has a specific receptor. Now these are kind of Y-shaped, but these are not antibodies, they're receptors. Remember at National 5 you learnt that hormones bind to receptors on the target tissues? In the same way, all cells have receptors to detect things. So these lymphocytes have specific receptors. Each lymphocyte has its own receptor, and that receptor is specific to only one type of antigen. So that means you need a lymphocyte, a different type of lymphocyte, for every possible pathogen that can invade your body. When a lymphocyte is activated, that means it's been selected by the antigen. Okay? And what happens is, when the receptor binds to the antigen, that lymphocyte starts to divide rapidly. And remember, one cell becomes two, becomes four. We call this population of identical lymphocytes a clonal selection process because they're all clones that are being selected for. They're all identical and the same. So let's say it's this lymphocyte there that has bound to that specific antigen. What's going to happen is that lymphocyte is going to divide to become 2, to become 4, become 8, and it's going to build up that clonal population. Okay, so that's what we mean by a clonal population. Now that's a good thing, because before you only had a handful of those specific lymphocytes in the body. It might take a while for one of them to come into contact with the antigen, and even if it does, there might be many pathogens in only a couple of lymphocytes. So they have to divide quickly to make this clonal population so they can effectively destroy the pathogen and prevent the pathogen from causing disease in the body. So I said there's two types of lymphocytes. They both work in the same way. They both have receptors on their surface that are stimulated when the receptor binds to an antigen, and the first thing they both do is to rapidly divide and create a clonal population. But there's also differences between the two. So we've got B lymphocytes and T lymphocytes. Let's look at B lymphocytes first of all. B lymphocytes produce antibodies. Now, these antibodies are also Y-shaped proteins, but they're released from the B lymphocyte, and then they travel and they attach onto the antigens on the surface of that specific pathogen. So there's the pathogen there, there's the antibodies approaching it. When they bind to the antigen, we call that the antigen-antibody complex. Now that deactivates the pathogen. That makes it harder for it to escape phagocytosis. So the B lymphocyte is triggered by a specific antigen. The B lymphocyte divides to give a clonal population of those specific B lymphocytes, and all of those B lymphocytes produce a specific antibody. Those antibodies attach to the antigens on the surface of the pathogen, forming an antigen-antibody complex, and then phagocytes come along and destroy those deactivated pathogens. And that's the process of the specific immune response by B lymphocytes. Now, B lymphocytes can sometimes respond to antigens which are actually harmless, and harmless is the key word. So they're not your own cells, they are foreign, but they don't cause disease. So for example, pollen can get into the body. If you've got hay fever, you'll know this, and it can cause what we call a hypersensitive reaction or an allergic reaction. And that is simply when the B lymphocytes recognise that harmless antigen, and it still triggers the response, and they still produce antibodies against that antigen when it's not needed. That's what we call an allergic reaction. Now, a way to remember this is B lymphocytes. Think about the B in antibody. So B lymphocytes create antibodies. If you think about bees, you'll think about pollen, and that's an example of an allergic reaction. But the key term is it's when B lymphocytes respond against harmless antigens. Okay, let's look at our T lymphocytes. Now, T lymphocytes' job is to destroy infected body cells. These are cells that a pathogen has gone into the cell. So it's not out travelling in the body, it's hiding inside the body cells. T lymphocytes induce something called apoptosis, which is programmed cell death. This is a series of events that you need to memorise. So, first of all, the T lymphocyte, it needs to know the cell's infected. So infected cells present the antigen of the pathogen inside of it on its surface. And again, the T lymphocyte has a specific receptor, and when it binds to that foreign antigen, that pathogen antigen in the surface of the infected cell, the T lymphocyte is going to rapidly divide, creating a clonal population of T lymphocytes. Now, how do they actually go about protecting the body? Well, the start of apoptosis always start with the T lymphocyte attaches onto the infected cell. So it physically attaches to the infected cell. The T lymphocyte then releases proteins, not the name of them, you don't have to know what they're called, just say the T lymphocyte releases proteins into the infected cell. Now, those proteins cause the infected cell itself to produce self-destructive enzymes, and that causes cell death. So the cell basically destroys itself. That's why it's called programmed cell death. And then the phagocytes come across, and phagocytosis removes the debris of that dead infected cell, as well along with the dead pathogens inside of it. What's key here is that they're called self-destructive enzymes because it's an infected cell itself that's producing them. Any wording that suggests the T lymphocyte is producing the self-destructive enzymes will cost you the mark. You have to be clear that after it attaches, it sends proteins into the infected cell, and those proteins cause the infected cell to start producing self-destructive enzymes, leading to apoptosis. Now again, this doesn't always work properly. When T lymphocytes don't work properly, we call this an autoimmune disease. So that's when, like the cartoon says there, the cell's trying to say, I'm one of your cells, it's one of your own cells. The T lymphocytes are being triggered by the proteins on the surface. They're seeing them as antigens that are foreign, and they're attacking it as if it's an infected cell when it's not. Okay, so T lymphocytes have to distinguish between antigens of your own cells, self-antigens, and of infected cells, non-self-antigens being presented on the surface. If they fail to do that, that will cause an autoimmune disease. Okay, so we talked about for both types of lymphocyte, the first thing that happens is the rapid division to create a clonal population. Out of that large clonal population, some of those cloned B and T lymphocytes are going to survive long term. Those cells that survive long term are referred to as being memory cells because they last so long. This means if you get a secondary exposure to the same antigen, you already have many cells in the body already. So that gives a more rapid rise to a new clonal population. Okay, so in the first example, we said there was one lymphocyte and it had to divide and create all the clonal population. If there's, say, 100 memory cells, then the first time they're activated, you'll get 200, 400, 800, 1600. So it's a much faster development of the clonal population. So the memory cells lead to a faster response, a faster clonal population. What's the point of this? Well, it means the pathogens are destroyed before they can cause the symptoms, before they can cause lots of disease and damage. Now, B lymphocytes have their own bit of extra information because B lymphocytes produce antibodies. So as well as the number of cells leading to a faster clonal population, as this graph shows you, when you have initial exposure, there's an increase in antibody production. Antibodies concentration increases. Compare that to the second exposure, there's two things that are greater. First of all, overall, there's a greater production. There's a higher concentration reached. Second of all, that line goes up much faster. It takes less time to get to the peak. So therefore, you can say that B lymphocytes, or memory B lymphocytes, will lead to a greater production and a faster production of antibodies upon a secondary exposure to the same antigen. Now, that doesn't mean that just as soon as you've been exposed to a pathogen, you're therefore immune to it. Even if that pathogen has triggered the immune response and led to memory cells, because some pathogens are clever, they have evolved this thing called antigenic variation. And that simply means they vary their antigens. And if you've varied your antigen's changed, then you're no longer specific to those memory cells receptors. So basically, memory cells will not recognise the pathogen after it's undergone antigenic variation. HIV is a virus that destroys T lymphocytes. Okay, so HIV is the actual virus molecule. When it's destroying your T lymphocytes and your T lymphocytes fall to a very low number, that's when you're actually diagnosed as having AIDS. And because you have AIDS, you've got such a low number of T lymphocytes, you're vulnerable to lots of different infections. And normally, if you die, it won't be the AIDS directly. It might be another infection like the flu that kills you because your immune system is so weakened. So in summary, you need to know that lymphocytes are specific because they have a specific receptor to an antigen. An antigen is any molecule that triggers an immune response. The antigens are found in the surface of the pathogens. B lymphocytes, and try and call them that rather than B cells, B lymphocytes produce antibodies which attach to the antigens in the pathogen, forming the antigen-antibody complex, and that deactivates the pathogen. T lymphocytes physically attach to infected cells. They diffuse proteins into the infected cell, and the infected cell then produces self-destructive enzymes and undergoes apoptosis. In both scenarios, phagocytes will then remove the dead cell. Now, if B lymphocytes respond to harmless antigens, that's an allergic reaction. If T lymphocytes don't regulate and they attack your own cells, that's an autoimmune disease. Memory cells are long-term surviving cells from your clonal population. They give a much faster response to a secondary exposure. In terms of antibody production, the antibody production is greater and it's faster. And that means that you'll normally, secondary exposure won't lead to disease because the immune response can destroy the pathogens before symptoms. Unless the pathogen is able to undergo antigenic variation, which means its antigens are changed and the memory cells will not recognise the new antigens, so you will not trigger that faster immune response. And finally, HIV destroys T lymphocytes, leading to a reduction in T lymphocyte number. Okay, you can pause this and go through at your own time. So let's try some questions here. Pause this, and when you're ready for the answer, press play. Okay, explain how a lymphocyte is able to recognise a particular pathogen. The lymphocyte has a receptor which is specific to the antigen on the surface of the pathogen. So keywords are receptor, specific, and antigen. Second part, T lymphocytes destroy infected cells by attaching to the infected cell, diffusing proteins into the infected cell, which causes the infected cell to produce self-destructive enzymes, leading to apoptosis. Now that's a full answer. That's only one mark, so I could probably say that T lymphocytes induce apoptosis in infected cells. But you never know with a marking scheme, so why not just give the full answer and show that you know it to the marker. Okay, again, pause this. Try and answer the question. It's 2 marks for each, and then I'll reveal the answers in a second. Okay, so for the first one, explain how a clonal population can be formed. There's 2 things to say. First of all, the receptors in the lymphocyte bind to the antigen, the 2 key words there. Second mark is given if you've said that leads to repeated division or it leads to cell division to form a clonal population. Second question, describe the role of phagocytes in the specific immune response. So again, it's not talking about 3.5 in phagocytosis. You're going to have to link your answer to the specific immune response. So mark 1 is if you said that phagocytes engulf pathogens after the antigen antibody complex is formed. Okay? Actually, let's cut this question out. This is old language. So ignore that marking scheme. Let me go back one and I'll tell you what you would get marks for. Okay, the role of phagocytes in specific immune response. If antibodies have formed an antigen-antibody complex and the pathogen is deactivated, a phagocyte will then engulf and destroy the pathogen. Full stop. I'm going to add some extra information, say, by using digestive enzymes stored in lysosomes. Now, that might not give me a second mark, so let's be safe and also say that after T lymphocytes have caused apoptosis of infected cells, phagocytes will engulf and destroy the remaining cell debris. Okay, so that's straight out the course notes how to answer that one. We'll ignore that old marking scheme. Okay, last one. Pause this. And for a condition, the type of white blood cell involved and how it's failed. So for this one, your condition is either an allergy. So we say that it's a response to a harmless antigen, or you could say an autoimmune disease, in which case you'd have to say the type of white blood cells, a T lymphocyte, and you have to say that it responds to self-antigens. Okay, so B lymphocytes respond to harmless antigens. T lymphocytes attack your own cells by responding to self-antigens.